as evidenced by the rapid development of the COVID vaccine. And now, I've done all of my training, and my primary interest lies in understanding how pathogens cause disease. Now, at the same time, I think the focus that we have on pathogenesis and the disease caused by pathogens sometimes hides the fact that by studying a pathogen and how it interacts with its host, we can uncover and see new general principles in biology that are relevant not only to pathogens, but the broader field of biology as a whole. So what I hope today, that through my seminar or talk, you'll get a feeling for the mechanisms by which pathogens survive in the host, but also see how studying pathogenesis can inform biology writ large. Also, I'd be happy to take questions that you have throughout the talk or at the end. So before I get started, I'd just like to thank the people who are really doing the work. I get to come and talk to you about science and all the great work that's being done in my laboratory, but it's really the graduate students, postdocs, and so forth in my laboratory that do all of the hard work day in and day out. So this is the current members of my laboratory here, and it's three former members, Kyle Grimm, Jacqueline Moret, and Euritzi Garcia, whose work I'm going to tell you about today. I'd be remiss not to mention our collaborators, both on campus in the Mining Microbial Genomes theme at the IGB, as well as across campus, and several international collaborators we have in Europe and Australia, as well as the people who provide the money to let us do all of this exciting work. Now, if we think about bacteria and bacterial pathogens in general, many bacteria will reside outside of our body where they really don't cause us any harm. But once they breach our skin or reach our insides, they can then go on to cause disease. So here we're just representing Staph aureus, which is a pathogenic microbe, but it's breached the skin and it's beginning to grow within our body. The presence of Staph aureus or other pathogens will lead to the recruitment of immune cells whose entire purpose in life is to eradicate invaders and prevent us from becoming sick. One of these cell types, or one of these immune cells, are neutrophils. Neutrophils will race to a site of infection, release a whole witch's brew of toxic compounds or antimicrobial peptides with the goal of killing and stopping the infection. One of the molecules or proteins that neutrophils release is a protein called calprotectin. Now, calprotectin does not directly attack the bacteria, but rather it scavenges the nutrients available to the bacteria. Specifically, it binds to transition metals such as manganese and zinc. This in turn results in a site of infection that is devoid of these nutrients, starving invaders from them. An example of this can be seen in the staphylococcal abscess. Here we have a tissue slice from an animal that has a staphylococcal infection. And here's the staphylococcal abscess. The staphylococcal abscess can be thought of as a pussy zit forming on your tissues and organs. Using a technique that allows us to produce a two-dimensional image of metal distribution within a tissue, we can see that the healthy tissue is replete in manganese and zinc as indicated by the warm colors, but the abscess itself is a highly metal restricted or metal deplete environment. So pathogens are coming in, they're being assaulted by the immune system. As part of that assault, the immune system is taking away these metals, the manganese, zinc, and others. Now, you might be thinking about this and saying, well, that's nice. Metals, who cares about them? What are they good for? Why do the bacteria care about them? Why does our body care about them? And the answer is, microbes in our body care about metals is because they're good for absolutely everything. Nearly every physiological process involves a metal in one form or another. Metals contribute to our ability to break down molecules, produce molecules, generate energy, or just general metabolism. Metals are essential for the function of ribosomes, which are essential for making new proteins. DNA and RNA synthesis, the heritable molecules of life, require metals for their synthesis, and then just resisting the general stresses that life encounters along the way requires metals. All told, about 30% of all proteins and 50% of all enzymes require metals for a function. So you can see that our body has a vested interest in keeping these critical nutrients away from pathogens, and pathogens in turn have a vested interest in trying to get these nutrients. Now, I just told you a minute ago that our body takes these nutrients away, rendering that site of infection virtually devoid of these metals. And so the question we can ask as people interested in studying molecular pathogenesis or bacterial pathogenesis is, how do pathogens overcome host-imposed metal starvation? What are the adaptations that bacteria have that allow them to thrive in this highly nutrient-poor environment? And there's two general strategies that bacteria can employ. One is they can work harder, increasing their ability to compete with the host for metals. And the other is they can work smarter, reducing their reliance on metals whose availability is restricted. And today what I'd like to do is tell you two stories, each which highlights one of the strategies that bacteria use to overcome this defense known as nutritional immunity. Now, every good story needs a protagonist or a villain, depending on your point of view. And the protagonist that we're going to talk about today is Staphylococcus aureus. Staphylococcus aureus is a bacterial pathogen that's gained a bit of notoriety because of its resistance to antibiotics, including methicillin, vancomycin, daptomycin, sort of current generation antibiotics. Staph aureus is rapidly becoming resistant to. This leads to increased morbidity and increased mortality or death. And so just for methicillin-resistant Staph aureus alone in the United States, there's approximately 300,000 infections every year, so about twice the population of Champaign-Urbana, and 10,000 deaths, or about a quarter of the university's population. All told, we see about $1.7 billion in an increased healthcare costs simply due to staphylococcal infection. So as someone interested in pathogenesis, I think it makes tremendous sense to understand the mechanisms by which Staph aureus causes disease, like as I said, so we can hopefully develop new treatments. Now, if we want to study metal limitation and how bacteria resist metal limitation, we need a tool or a mechanism to impose metal limitation on the bacteria. Now, what we've decided to do is take a book or a page out of the host's playbook and use the same molecule the host uses to impose metal starvation during infection, to impose metal starvation in a test tube or in culture. So you'll recall I told you there was a protein called calprotectin that binds manganese and zinc and will starve pathogens for these metals during infection. We've developed the ability to produce calprotectin in culture, allowing us to add it to cultures in vitro or outside of an animal and impose metal starvation on pathogens, allowing us to study how they respond. And here I'm just showing you that addition of calprotectin inhibits the growth of a wide variety of pathogens via metal starvation. So we have Staph aureus, two closely related sister species, Epidermidis and Staphylococcus, the Enterococci, Pseudomonas aeruginosa, Acinetobacter baumannii, Shigella flexneri, and E. coli. And nearly every pathogen that that has been exposed to calprotectin, it's been seen that it will inhibit their growth via starving them for metals, giving us a really good tool to ask, okay, what are the adaptations that they're using to survive in a metal-limited environment? Now, we can go back to that struggle for metals. We can think about what's going on when Staph aureus first arrives in the host. We have Staph aureus over here, and it encounters an environment where manganese and zinc are present. After this, the host response is going to start. Calprotectin is going to be released to that site of infection, and it's going to pull manganese and zinc away from the bacteria, taking it away from the site of infection. Now, if we think of this as a tug-of-war, if somebody's trying to pull something away from you, you can turn around and pull back harder, trying to bring it to yourself. And that's exactly what Staph aureus does. It tries to pull metals towards itself and incorporate them into its cytoplasm using proteins called transporters. Now, through some prior work, we knew that Staph aureus expresses two transporters for manganese, illustrated here, and we then went on to show that these transporters are important for the ability of Staph aureus to cause disease and important to resist the antimicrobial effects of calprotectin. Now, these studies told us about how it resists manganese, but it left us with a hole in our knowledge regarding how Staph aureus competed for zinc. And when we started trying to understand how Staph aureus competed for zinc, we ran into a problem. And that problem specifically was the transporters that Staph aureus uses to obtain zinc were unknown. Now, while they were unknown, that doesn't mean we have nowhere to start with. There's other bacteria in which zinc transporters have been characterized, and one of the most widely distributed zinc transporters in other bacteria is present in Staph aureus. This is a system known as the ADC-ABC system. And in other bacteria, what it does is it transports the zinc from the extracellular space into the cytoplasm so bacteria can use it. So we started with the hypothesis that perhaps the ADC system is how Staph aureus is competing with calprotectin in the host for zinc. So we asked, is ABC necessary for Staph aureus to resist zinc starvation? Now, Kyle, the graduate student I mentioned a few minutes ago, went off eager to tackle this problem. He worked on it for a while, and he came back and he said, no, the ADC system is not necessary for Staph aureus to resist zinc starvation. I turned around to Kyle and said, are you really sure? Are there any more experiments that you could do? And he says, well, maybe one or two. He went back, did those experiments, came back to me, and his answer was still no. And so we're left with a conundrum. ADC-ABC is the primary transporter, the known transporter that bacteria express to acquire zinc. We know that zinc acquisition is absolutely essential for bacteria to survive. Yet the one potential zinc transporter we could identify wasn't important for resisting zinc starvation in the host. So we had a bit of a conundrum. So we had to take a step back and think about, well, how could we identify a priori the zinc transporter expressed by Staph aureus? Now, if we think about transporters and we think about how bacteria respond to metal availability, there's a general strategy that bacteria and microbes use to control the expression of metal uptake systems. They use proteins known as regulators, and in the presence of metals, or when metals are abundant, that regulator will bind to the metal, and that the binding of the metal, or excuse me, the binding of the metal to the protein will then lead to repression of gene expression. You can think of this as, well, I'm full. I don't need to go eat more food, so you're not going to reach out and grab more food. However, when you're hungry or the substrate or metals are absent, the metal is no longer going to bind to the protein. That protein is then going to no longer repress the expression of metal transporters, and therefore the bacteria are going to reach out and grab and import metals into the cell. So we can take advantage of this regulatory scheme, and we can grow bacteria in the presence of metals, a situation where the transporters should be off. But by creating a mutant in the bacteria, we can eliminate their ability to regulate or repress the expression of their metal importers. And so what's going to happen is we'll have bacteria grown in the presence of metals, but because the regulatory protein is missing, they will overexpress their metal transporters. So we did this experiment. We eliminated the protein that regulates the ability of Staph aureus to control zinc homeostasis. It's a protein called ZR, and we identified two potential transporters. One of these you're going to recognize was this ADC system or ADCA protein, and we see that when you eliminate ZR, the zinc uptake regulator, expression of the ADC system increases as we would expect. We also saw that the CNT system, another system that has homology to a transporter but wasn't known to bind or import metals, also is increased in a ZR mutant. However, if we eliminate the other metalloregulators in Staph aureus, MNTR, which regulates manganese uptake, and FR, which regulates iron uptake, we see no increase in either the ADCA or CNT system, suggesting that these are likely to be the two zinc transporters possessed by Staph aureus. So we now have somewhere to start looking again. And so we can ask, is CNT ABCDF, or this transporter here, necessary for Staph aureus to resist zinc starvation? We already know that the ADC system is not necessary. What we did is we once again took advantage of our ability to make mutants in bacteria, our ability to eliminate genes and therefore prevent them from expressing the gene and the associated protein. So we have wild type bacteria here in orange. We have a strain that lacks the ADC system in blue, a strain that lacks the CNT system in green, and a strain that lacks both of these transport systems in purple. And what we did is we grew the bacteria at increasing concentrations of calprotectin and just measured their growth. So the higher the value on the y-axis here, the better the bacteria grew. And we can see in orange and blue that these two strains grow the same across all the conditions we tested, telling us once again that the ADC system is not necessary for Staph aureus to resist metal starvation by the host. However, if we look at this green bar here, we see that as we increase the concentration of calprotectin, a strain lacking the CNT system grows worse, meaning it's not able to compete with calprotectin for zinc or survive calprotectin-imposed zinc starvation. Now then, if we look at the strain that lacks both the ADC system and the CNT system, we see that at very low concentrations of calprotectin, this strain has a defect, telling us that while the ADC system by itself is not necessary for to resist zinc starvation, it will contribute to the ability of Staph aureus to acquire zinc if the CNT system is missing. Now, this is in culture. This is in a test tube. Yes, we've taken calprotectin, the molecule that imposes zinc starvation, and use that molecule to ask how are they growing. But a test tube does not fully recapitulate what's going on during infection in a live animal or in us. And so we turn to infection modeling to ask if these systems truly are important for the ability of Staph aureus to cause infection. And we do this by infecting mice and evaluating the number of bacteria in each tissue. If the bacteria are doing well, we would expect high numbers of bacteria in the tissues. If the bacteria are doing worse, we would expect lower numbers of bacteria in each tissue. So we can see here that when we infect mice with wild type bacteria, we get about 6 logs of bacteria. If we infect with that ADCA mutant, we get a similar approximately 6 logs of bacteria. But if we infect with the CNTA mutant, so a strain that doesn't have the CNT system, we see a reduction in bacterial burdens, indicating that the CNT system is the primary system used by Staph aureus to acquire zinc during infection. And then consistent with our in culture observations, if you eliminate both the CNT and ADC systems, we see a further reduction in staphylococcal virulence. So now we can answer the question, how does Staph aureus compete with the host for manganese and zinc? We now know there's 2 manganese transporters and 2 zinc transporters that Staph aureus is going to use. Now, this was all very exciting. We've identified a new zinc transporter, But looking at the genomic structure where this transporter was encoded, we came to realize that this probably wasn't the end of the story or didn't tell the full story. And that's because the CNT transporter is encoded in a region of the chromosome that encodes not only for a transporter, but 3 biosynthetic genes and an efflux pump. These biosynthetic genes, someone had previously shown, encoded for proteins that produced a small molecule capable of binding a wide variety of metals very tightly. And this efflux pump here is responsible for secreting this small molecule known as staphylopine into the extracellular space. And so we began to wonder if perhaps the CNT transporter was functioning with staphylopine to bring metals back into the cell, specifically zinc. So the first thing we did is we made mutants, or strains that lacked the synthesis genes or the efflux pump, and asked how well they could survive in the presence of calprotectin or grow in the presence of calprotectin. And just like the CNTA mutant or the strain that lacks the transporter, we see that these mutants also have a growth defect in the presence of calprotectin. Now, this tells us that they are important for resisting metal starvation, but it doesn't actually tell us that they're working with the CNT transporter to bring metals into the cell. To do this, we took advantage, to test this, we took advantage of the fact that staphylopine is a metallophore that is secreted into the supernatant. It's secreted into the environment where bacteria are growing. So if you harvest supernatant from bacteria that can make staphylopine or from bacteria that cannot make staphylopine, you can combine that with some genetic tricks to ask, is staphylopine working in concert with the transporter? So what we did is we grew wild-type bacteria and a strain that cannot import staphylopine. This is the CNTA mutant here in blue, and a strain that cannot make staphylopine here in green. If we supplement these bacteria with the supernatant that's harvested from bacteria that cannot make staphylopine, or from a CNT-KLM mutant, there's no staphylopine present, and we see that the only strain capable of growing is the wild-type bacteria, which can both produce its own staphylopine and import staphylopine. However, if we use supernatant harvested from either a CNTA mutant or wild-type bacteria, both of which can produce staphylopine, we see once again that wild-type bacteria grow. We see that our CNT-KLM mutant, or a strain that cannot synthesize staphylopine, can now grow. But we see that the strain that cannot use staphylopine, or conceptually cannot use staphylopine, does not grow, establishing that the CNT transporter works in concert with staphylopine to bring metals, and specifically zinc, into the cell. So we can now revise our model. We can say that the CNT staphylopine system is responsible for competing with zinc, and that staphylopine is a zincophore, a small molecule produced by bacteria that is secreted and then brought back into the cell with metals. This was a particularly exciting finding because bacteria were known to produce small molecules to obtain metals such as iron, but it wasn't clear and it wasn't known that bacteria would produce small molecules to obtain other non-iron metals. And this was one of the first demonstrated examples of that. Now, I told you at the beginning of this talk that by studying pathogenesis, we could learn about biology writ large, yet I've told you a story so far that's only focused on Staph aureus. And what I want to turn to now is some work that we conducted with the IGB and with the Mining Microbial Genomes theme to ask, well, how widely distributed are these bacterial metallophores that are capable of importing zinc, or capable of bringing zinc into bacteria? So here we have a cluster, and each one of these dots represents a genetic loci capable, we think, of making staphylopine. These This cluster here largely represents bacteria that are closely related to Staph aureus. So they're going to be found on us, on animals, other warm-blooded hosts. We also see that the Yersinia species of bacteria or genre of bacteria will make a molecule similar to staphylopine called Yersinia-pine. Yersinia, these bacteria include Yersinia pestis, the causative agent of plague. We can look at that and say, well, those are both groups of bacteria that are pathogens. What about these broader non-pathogenic bacteria? And then we can see in this that other bacteria, these non-pathogens, begin to make metals as well. We see that the Pseudomonads, including Pseudomonas aeruginosa, but also other Pseudomonas species, a common organism found in the dirt and on plants, make a molecule called pseudopalene, which is similar to staphylopine. And then we can look at this group over here, and we can see that many bacilli species and actinobacteria, also organisms that grow and live in the soil, water, and so forth, will produce these zinc scavenging molecules, telling us that showing that by studying zinc acquisition in a pathogen, we've been able to identify a zinc uptake system that's present in both pathogenic and non-pathogenic microbes that inhabit a wide variety of different ecological niches. So we think the C&T type system is a new high affinity zinc uptake system expressed by bacteria widely. So I've told you this. Maybe I'll stop here for a moment and ask, do you guys have any questions about what I've shown you so far? Okay, not seeing any questions, I'll move on. But if you do have questions later on, please feel free to ask them. So the next thing I'd like to turn to is working smarter. I've told you how they're going to work harder, how they're going to work hard to obtain metals, but now I want to tell you about how bacteria work smarter. And specifically, I want to focus on the role of metals in oxidative stress defense. One of the tools or weapons that neutrophils and other immune cells have to combat pathogens and non-pathogens alike is the production of reactive oxygen species which cause oxidative stress. One of these reactive oxygen species is superoxide dismutase. Bacteria and other forms of life protect themselves against superoxide by expressing an enzyme called superoxide dismutase. Superoxide dismutase requires a metal for function. And we can see how this superoxide defense and calprotectin defense of the host come together here, in that when we grow bacteria, specifically Staph aureus, in media or metal replete media, we can see some appreciable amount of superoxide dismutase activity. However, when we treat the bacteria with calprotectin, which is going to remove the metals from the media, we see that superoxide dismutase activity is reduced. So nutritional immunity conceptually can, or does, I should say, inhibit the bacterial defenses against superoxide, a second weapon that the host is using to combat invaders. So we know that host-imposed manganese starvation inhibits staphylococcal SOD activity, and we know that it sensitizes Staph aureus to oxidative stress. We now know that this happens both in culture and within an animal during infection. Now, this makes sense. We know that calprotectin binds manganese and zinc and starves bacteria for these metals. We also know that Staph aureus possesses two superoxide dismutases, which at the time we were beginning these studies were thought to use manganese. So we have SodA, a manganese-dependent enzyme, and SodM, a manganese-dependent enzyme. So we can envision calprotectin binds manganese, it starves the cell for manganese. These enzymes lose activity because they are reliant on manganese for function. However, when we delve deeper into how calprotectin was affecting superoxide dismutase activity and the ability of Staph aureus to resist superoxide, we came across a really unexpected finding. Now, similar to the previous experiment I showed you, we've created a series of mutants, except this time we've eliminated the different superoxide dismutases that Staph aureus expresses. If we grow Staph aureus in a metal-replete environment, we see that loss of SodA in green here results in a growth defect. However, as we increase the concentration of calprotectin, we see that this growth defect relative to wild-type bacteria disappears. However, if we look at the SodM strain, the strain that lacks SodM, we see that in the absence of calprotectin, that mutant has no growth defect. However, as we increase the concentration of calprotectin, it now begins to have a growth defect, indicating to us that SodM, despite reportedly being a manganese-dependent enzyme, is necessary for Staph aureus to resist manganese starvation. So we have a new question. How does SodM, a manganese-dependent enzyme, promote resistance to manganese starvation? It's hard to envision how taking away an enzyme that the metal that an enzyme uses for activity would allow that enzyme to contribute to surviving starvation. How does that work? And didn't add up to us. And told us that what that tells us is that we don't fully understand what's going on with the staphylococcal superoxide dismutases. And so we undertook a series of studies to understand what's going on with SodA and SodM. How is SodM promoting resistance? Now, before I jump into those studies, I just want to talk about superoxide dismutases writ large. Superoxide dismutases are one of the most widely distributed enzyme families or enzyme groups in the world. Basically, all forms of life on the planet express a superoxide dismutase in one form or another. We typically, when talking about superoxide dismutases, classify them by the metal that they use. We have enzymes that require iron, we have enzymes that require manganese, and we have enzymes that require copper and zinc. Now, from a functional perspective, this is a nice way to talk about them, but it hides the fact that the iron and manganese superoxide dismutases are basically the same protein. They're the same protein superfamily. They have identical structural folds when you look at their structure or solve their crystal structures. And it hides the fact that we cannot actually predict a priori when you find a superoxide dismutase or manganese/iron superoxide dismutase in a genome, what metal it is going to use. So you can find an enzyme that will look like an iron and or manganese dependent superoxide dismutase, but because we don't understand how these enzymes work or what drives specificity, we can't predict without doing experiments what metal they're going to use. In light of this, we thought, well, maybe we just got the characterization of SodA and SodM wrong. I mean, why would an organism make two manganese-dependent enzymes anyways? It'd make a lot more sense if SodM used a different metal. And so what we did is we purified SodA and SodM and selectively loaded them with different metals and measured their enzymatic activity. And so up here, we loaded the enzymes with iron, and down here, we loaded the enzymes with manganese. The red lines shows you the trace associated with iron. As it goes higher, there's more iron. The blue line shows you the trace associated with manganese. Once again, as it goes higher, it means there's more metal. And in lines you can't see because they're hiding behind the metal, that just shows us where the protein is. And we can see here that we can selectively load SodA and SodM with either iron or manganese, and then we can then assess their activity. As we would expect from a manganese-dependent enzyme, SodA had negligible activity with iron and robust activity with manganese. However, when we looked at SodM, we saw something unexpected. We saw activity with iron, and we saw activity with manganese. And this is just something that was surprising and confusing at first, because the dogma in the field is that you have manganese-dependent variants of superoxide dismutase and iron-dependent variants of superoxide dismutase, and nothing in between. You don't have enzymes that can use both. And this view and belief came about for several different reasons that accrued over time. And I'm not going to go into those reasons today. But if you go back in the literature and you just look at some of the initial studies on superoxide dismutases, you found an additional type of iron manganese superoxide dismutase, and these were cambialistic enzymes. And these enzymes have the ability to use either iron or manganese. But because of their lower ability to use iron and or manganese, their importance was or the importance of cambialism was generally dismissed to biological situations. So we now know that SodM can use both iron and manganese, and so we began to wonder if maybe the ability of SodM to use iron is what it's allowing it to contribute to Staph aureus's growth when manganese starved. So what we did is we grew Staph aureus once again in the presence of increasing concentrations of calprotectin, and we lysed open the cells and did an assay that allows us to visualize the superoxide dismutase activity associated with either SodA, SodM, or a heterodimer formed by both SodA and SodM. We can see here that as we increase the concentration of calprotectin, the activity of SodA, that manganese-dependent enzyme, goes down, and the activity of SodM, that cambialistic enzyme, goes up. But this doesn't tell us if this activity is dependent on manganese or dependent on iron. So we treated the gels with hydrogen peroxide, which kills iron-dependent enzymes, and we can see that the activity associated with SodM goes away, telling us that in the presence of calprotectin, SodM is becoming loaded with iron, allowing it to maintain a defense against oxidative stress, promoting growth when the bacteria are manganese starved. We went on to show that this happens in vivo as well. So we now know that the ability of SodM to use iron allows it to maintain a defense against oxidative stress when manganese starved. And so from these studies, we've really rediscovered, if you will, that superoxide dismutases of the iron manganese family shouldn't be thought of as these distinct separate enzymes, but really should be represented as more of a continuum where you have some enzymes that truly are manganese specific, and you have some enzymes that are truly iron specific, but a whole host of these enzymes occupy some space in between where they have differing abilities to use iron and or manganese. And we think that this false dichotomy that you have to be either iron dependent or manganese dependent has really hampered our ability to understand the mechanisms or the underpinnings of why some enzymes are using iron and some enzymes are using manganese. Importantly, this family of proteins is present not only in bacteria, but also archaea, as well as eukaryotes. So we see them and superoxide dismutases are present in this family of superoxide dismutases are present in all plants, all people, basically all eukaryotic microbes. So this family is widely distributed. And so these studies have really informed how our understanding of these enzymes across a broad swath of biology. Now, if this was an infomercial on late at night, which I will admit to watching when I cannot sleep, I would say, but wait, there's more. And while it's not an infomercial, I am going to say, but wait, there's more. And that's because, recall I told you, we could not predict where an enzyme fell in terms of using iron or manganese because we didn't understand the molecular features that dictated that specificity. We didn't know these specific amino acids or the sequence that said, hey, you should use manganese. Hey, you should use iron. And the reason, despite 40 years of study, people have had trouble deciphering this is because while the superoxide dismutase family has a highly conserved structure overall, there's very low levels of sequence identity. So this means that the proteins, when you fold them all up, roughly look the same. But if you look at the actual sequence or the order of the amino acids in all of these proteins, They're highly divergent and very different. To put this in perspective, we have some enzymes that use manganese, and they're at best 50% identical. We can take another set of enzymes, say one that uses iron and one that uses manganese, and they'll be at best 50% identical. So there's relatively low levels of homology preventing us from comparing different enzymes and asking, hey, what are the important features? This doesn't actually happen in the staphylococcal superoxide dismutases, and that's because SodA and SodM are 75% identical, with the majority of the differences occurring at the surface of the protein, which is unlikely to contribute to metal selectivity or its activity with different metals. So we solve the structure of SodA and SodM with different metals, specifically iron and manganese. And what we notice is that there are 3 residues within proximity to the active site, which you can see here, residue 19, 160, and 159, which differ between SodA and SodM. Now, if these residues are important for controlling the metal specificity or the ability of this enzyme to use a metal, we would expect that if we swapped them, we should change the activities of these enzymes. So we should be able to take SodA, a manganese-dependent enzyme, and make it more cambialistic. And conversely, we should be able to take SodM and convert it into a more manganese-dependent enzyme. So we went ahead and created a series of mutants in these positions where we reciprocally swapped the residue from SodA into SodM and the residue from SodM into SodA. And we assessed the activity of these proteins with both manganese and iron, as we did previously for the wild type proteins. And what we can see here is that as we mutate these residues or swap these residues, we can convert SodA from being a primarily manganese-dependent enzyme here in purple to a primarily cambialistic enzyme here in green. Conversely, we can convert SodM from a primarily cambialistic enzyme over here into a primarily manganese-dependent enzyme over here. Now, clearly, these 3 residues do not explain all of the metal specificity and these enzymes, but I think they highlight how by studying a pathogen, we can gain greater insights into a broadly relevant biological process. So what I'd like to leave you with today is that our bodies are a nutrient-restricted environment, and this nutrient restriction forces bacteria, both pathogens and non-pathogens, to adapt to it. And it's the ability of pathogens to adapt to that nutrient restriction, which allows them to cause disease. Bacteria have numerous strategies by which they compete with the host for metals, so numerous mechanisms by which they're going to pull the metals toward them. And we now know that bacteria are going to replace proteins that are inactivated by nutritional immunity with those that will remain active. So SodA is inactivated by nutritional immunity. Staph aureus then produces SodM, which can retain activity. And finally, by studying how pathogens adapt to the host, how they adapt to the stresses encountered within the host, we can gain a greater understanding of how but just how life writ large adapts to stresses such as metal starvation or oxidative stress. With that, I'd like to thank once again all of the people in my group, all of my collaborators, people who provided the funding, and take any questions that you may have.
Yeah, thank you very much, Dr. Kehl-Fie. If you have any questions, everyone feel free to put them in the chat or just talk to us. I did get us started, have a couple of questions that popped up. So how does metal starvation affect the host tissues in these things? So if the hosts are in the same scenarios?
Yeah, so that's a good question. And so I think we have to think about this in 2 ways. One of this is growth. So if we think about bacteria, you're going to have a few bacteria that will initially start an infection. And those bacteria are going to grow rapidly. They're going to be dividing. They're going to be making more bacteria. So they're going to have to increase their biomass. And every time a bacteria divides, it has to repopulate the cell with metals again, because every division is cutting it in half. And so a bacteria that's rapidly growing and rapidly dividing needs to actively be bringing metals into the cell. Our body and our cells definitely need metals, absolutely 100%. There's numerous genetic diseases that or defects in metal homeostasis, metal uptake lead to illness. But as a function of time scale, our cells, or the cells of our body, grow and divide much more slowly. And so the bacteria are dividing every 20, 30 minutes. Our cells are dividing over the course of days and weeks. And so because our cells are dividing slower, they're going to have less of a need to acquire more metals or nutrients, just because they grow more slowly. The other thing to think about is sort of a what problem do you want to deal with first? At a site of infection, our body, yes, we'd like not to have rampant tissue destruction and so forth, but it's a higher priority to eradicate the bacteria than it is to make sure all the cells are getting all the nutrients that they need. If you recall, I mentioned that the staphylococcal abscess is functionally a pussy zit, and that pus and that goo is the bacteria. It's also a large collection of immune cells whose whole purpose is to rush to that site of infection, release all these toxic molecules, and kill the bacteria. And so they're going there, those cells are dying, but that's what their job is to do. And so there's going to be tissue damage associated with infection, but it's better a little tissue damage than a raging infection, if you will. Hopefully that addresses your question.
Yeah. And then the other question that I had, you also found these similar zinc binding proteins in a lot of non-pathogenic bacteria. What scenarios, because like they're not going to be necessarily competing with the host protein. So what scenarios do those non-pathogenic bacteria use?
Yeah, so that's a good question. So from a pathogenic, if we think about things from a pathogenic perspective, we say, oh yeah, the host does all of this stuff. But if we get out into the broader world, we can't just assume that nutrients are freely available. For example, seawater. We not think about seawater a lot. We think, oh, there's some salt in seawater, right? It's salt water. But salt water itself is actually a fairly nutrient-poor environment. And transition metals such as zinc are actually in fairly low abundance. You can say, well, what about dirt? Dirt, there's a lot of stuff in dirt. There's a lot of nutrients in dirt. Well, not only is there nutrients in dirt, but there's also a lot of other bacteria and other microbes in dirt. And so while the microbes that produce these metal uptake systems may not be in a per se zinc limited environment because the total abundance of zinc is reduced, but they're competing with all of their neighbors. They're jostling with all the other different species of bacteria that are trying to grow in this environment. And whoever gets that zinc first is going to have an advantage when it comes to growing. And so some of these systems may not provide an advantage to them in the context of a host microbe interaction, but it may provide an advantage in a microbe situation where, because of the diversity of microbes in an environment, they're competing with all these other species for zinc.
Thanks. We did have another question on the YouTube channel about are there any insights into abiogenesis?
Can you repeat that question?
Yeah, do you have any insights into abiogenesis?
I'm not quite sure what the question is referring to. So if they want to clarify, I'd be happy to try and answer that.
So yeah, if you have that question, feel free to try to clarify that. Otherwise, if you're you're welcome to unmute and talk. Otherwise, if there's any other questions, feel free to let us know.
Thank you all for attending as a scientist, and it's always a great opportunity, a great pleasure to have the opportunity to talk to people about the work that we're doing and all of the fantastic studies that the people in my laboratory are undertaking.
And yeah, thank you so much for presenting your work. It's always super cool to see how these studies in other species can impact research in our own bodies and on other different animals, different organisms, how closely related all of these different systems are. All right, well, I don't see any responses from the chat there. So if you have, oh, David, did you have a question? I can't quite hear you, David. Well, I guess not, but feel free to, any other questions that get submitted to us either in the chat or you can add it to our Facebook channel. And then I'm happy to get answers to any other questions we receive. But thank you so much for presenting your work with us.
Well, thank you all for attending, and I wish everyone safe travels.